you see the screen?
Yes, sir. Yes, sir.
Did you
It's
see
not full screen yet.
It's not full screen yet, is it?
Yes, yes.
Okay, let me share it again because it's doing
Okay.
some funny. It's full screen for me now, and yeah. Now is it better?
Yes, sir.
Yes.
Okay, all right. So good afternoon, everyone. My name is Nikhil Patkar, and I hope we have a great session today. I'm going to talk on the approach to mutation testing. So I'm going to talk a little bit about everybody's talk and talk bits and pieces from what people are going to say. But the general idea is that before we delve into each and every one of these talks in detail, the first idea would be to just give a broad overview about how molecular pathology is actually applied in clinical medicine and how do we do different kinds of tests. So first is introduction to the genome and the basic different types of PCR that are there. So cells, if you see how human beings, how humans develop, cells that develop into gametes are called as germline, and these cells are typically haploid, and they have half the number of chromosomes. Excuse me, sorry. So they have half the number of chromosomes. So the cells that contribute to one's body are called as somatic cells, and typically they are formed by a union of two gametes, and they are diploid, and therefore 23 plus 23 makes 46 chromosomes, which are there. Another unique aspect is that you also have mitochondrial chromosomes, which are exclusively present in mitochondrial genome, which are inherited from the maternal side. So these cells, if you look very deeply into the structure of these cells, you can see that every cell has cytoplasm and it has a nucleus. Now this nucleus in interphase has this chromatin which is wound around like this thread or a ball of thread. And if you zoom into it, you can see that this is further organized like a rope, like a rope which is called as a solenoid. And if you look further into it, it essentially comprises of a strand of DNA which is wound around the wall, around this ball of histone. So this is the essential structure of nucleic acid, which is tightly wound around the nucleus. Now, if you look into each DNA strand, you can see that it is double-stranded, and you have a top strand and you have a complementary strand. But when you look at the reference genome, or you look at any data which is present from the genome, you can see that this genomic data is represented as a single strand because the sequence which is present on the opposite strand can then be inferred based upon the double-stranded nature of the genome. When we start using genomic technologies or when you start reading genetic literature, you come across certain terminologies like what is a locus or what is an allele. So a locus, or the plural is loci, are segments of DNA which occupy a particular region or location on a chromosome. A locus can be large, for example, the HLA locus, or very small, for example, the beta globin gene locus. So an allele is an alternative version of DNA sequence which are present at a locus. So for example, this is an allele for short hair in a dog, and this is allele for long hair, or allele for white fur, and allele for black fur. And this region is known as the locus or coat length or the coat color. So this is how this terminologies are divided. I please request the rest of the people to mute themselves so I can hear some background noise. So there is also some other terminologies which are used in the genome, which are wild type and mutant. The wild type allele is the single prevailing allele which is present in more than half the individuals in the population, and it is said to be the common allele. So the wild type allele is commonly present in people, and the mutant allele are versions of that particular allele which differ from the wild type allele because there is the presence of a mutation. So you can see that the wild type hemoglobin here is CTC, which predicts for glutamine, and if it is changed to CAC, you have a sickle cell hemoglobin, which is valine, so which is glutamine changing to valine. So if there are two or more relatively common alleles at a allelic frequency of more than 1% locus in the population, the locus is said to exhibit polymorphism, or it literally means many forms in that population. A private allele, sometimes there are variations, but they are not frequent enough to be considered to be polymorphisms. For example, if you have a variation within one particular community in India, or for example, if there is one particular community which expresses a particular polymorphism in one caste or one community, that is said to be a private allele. There is also the concept of haplotype, which is a set of DNA variations adjacent to one another at the same locus, which tend to be inherited together. So this, I don't know why this is doing this. There is somebody else who says to have shared the screen.
SP, one second, I'll just check something in the system.
Please, sir, share your screen.
No, who is this? SP is pressing the share.
Yes, yes,
We have to.
now you can share yourself, PPT.
Yeah, I know. I already did that
Yes,
twice.
I made you present.
Okay, so another set of DNA variations which are adjacent to one another at the same locus, which are inherited together, are called as haplotypes. And when haplotypes are this particular set of alleles are linked together, they are called as linked polymorphisms, based on which we do linkage analysis. So let us look at a group of individuals here. Now, at this particular locus, you have A or G. So these are the two different types of alleles that are there. But this A, G, and T are always inherited together in one individual, whereas G, T, and A are inherited in another one, or an A, G, and A are inherited in another one. And these are called as different haplotypes. So a mutation is a permanent change in nucleotide sequence or arrangement of DNA. The classification of mutations is typically done by the size of alteration. So even chromosomal abnormalities are called mutations, but in the colloquial usage, you don't refer to them as such. Chromosome mutations can be changed in the number of chromosomes. You can have regional or sub-chromosomal mutations, for example, changes involving a portion of chromosome. You can have structural rearrangements which involve one or different parts of chromosomes, or you can have gene or DNA mutations which involve substitutions, deletions, or insertions of DNA. So we can show some examples of each of these things. So genomic alterations are classified as microscopic or submicroscopic. So when you say microscopic, they can be detected through chromosome banding techniques, which are typically done by cytogenetics. And for it to be detected by cytogenetics or metaphase analysis, it has to at least be the alteration has to be at least 500 kb big enough. Submicroscopic variations are variations which are beyond the resolution of conventional karyotyping, and they are identified through techniques such as RACGH or NGS. All right. So you can see these are examples of microscopic variations. You have whole chromosome aneuploidy. For example, there is this is one chromosome, and here you can see there is a gain of another chromosome. For example, you have a chromosome for trisomy 21. You can also have loss of a chromosome which is there, which also is a part of whole chromosome aneuploidy. You can have segmental aneuploidy, in which deletion of the part of a chromosome is lost, for example, monosomy 5 or monosomy 7, which are seen in myeloid malignancies. There is also translocation of another chromosome, wherein one chromosome gets fused to this chromosome, for example, BCR-ABL or 921, 821, or you can have duplication of part of chromosome, wherein one region of the chromosome gets duplicated. What do you mean by when you say gene mutation? So let's look at this hypothetical gene. This is the coding region of the gene, and you have the reference sequence on top. As you know, 3 bases come together to form or code one amino acid. When one particular base here, for example, GCA is changed to, for example, TCA changes to GCA, you have serine changing to alanine. So this is an example of a base substitution. Sometimes you can have a deletion of one or more bases or insertion of one or more bases. These changes, when they are not in replicates of 3 or when they are single or double or multiple lines, they can cause something known as frame shift mutation. So let us see what they are. So let's look at this line in English, I love Guns N' Roses in a rock concert. If you remove the and Roses part of it, it says I love guns in a rock concert. Now the meaning of these two sentences is completely changed. The same analogy can be used in the molecular biology context, wherein the incorrect amino acid follows for changes. For example, because the GCA, the G is deleted, now you have CAC, which forms the codon, and CGT, which forms the next codon, and so on. So the entire meaning or the entire translation of amino acids in the protein, therein below the downstream of this change changes to the premature introduction of a stop codon. So not only is the amino acid sequence or the protein constituent changing, but there is also an abrupt synthesis or termination of synthesis of amino acids. So there are also deletions can be like you have A, B, C, D, E. One particular region is deleted. You can have duplication wherein one particular region gets duplicated. You can have inversion type of abnormalities wherein the entire region gets inverted. A common example of this would be mutations that are seen in hemophilia type A. So I'm going to just briefly talk about what goes into a polymerase chain reaction. I understand this will be also talked about by some of my other colleagues, but it's important to understand a little bit about this. So every PCR reaction has a template nucleic acid, which is a DNA or a cDNA. You can have you always have primers, which are stretch of nucleotides, which are complementary or reverse complementary to the region of interest. They are usually 2 in number. They can be more, but they are never less. And you also have DNTPs, such as ATGC, which will use these DNTPs to form new DNA by putting these DNTPs ahead of the primers. You also have Taq polymerase, which is the enzyme with DNA replicating properties, which is resistant to high temperatures and uses these DNTPs to synthesize the new DNA in the PCR reaction, and you have buffers. All right. So in one PCR reaction, the first step is denaturation, wherein at high temperatures, the DNA strand breaks open to form 2 separate strands. So these strands are typically held apart by hydrogen bonds, and at high temperature, these bonds break. And then the temperature cools down, and the oligonucleotides or annealing of these primers takes place to the corresponding region of the genome, both on the upper strand as well as the complementary strand. And then using Taq polymerase, these DNTPs help synthesize and form new nucleic acid like this. And in this manner, one DNA copy becomes 2. But it's not just 2 cycles. This cycling is done through multiple cycles, so that you can have exponential increase in these the amount of template copies. So the first application of PCR to molecular medicine is to amplify nucleic acid which does not belong in that environment. So when you say environment, it can be the human sample, it can be a soil sample or a food sample. What's important is what is the appropriate clinical context and the patient and the symptoms to which what is there for the patient. So typically, an example of this would be detection of a pathogen in a focus of infection, for example, tuberculosis, or in a oral swab SARS-CoV-2 or hepatitis C from a blood sample. So it will confirm the absence or presence of a particular pathogen. So why do we need multiplex PCR? So let us look at this particular scenario where a patient is sick, he has meningitis, and you have a very tiny amount of sample, typically half an ml of CSF, which is provided to you. Now the number of organisms that can cause meningitis are so many. So are you going to use separate PCRs for each of these? One, it is very expensive. 2, you will also run out of sample here. So when you want to do tests like this, you must try to optimize the usage of sample and maximize the testing that can be done. So a way in which you want to do this is by multiplex PCR, in which you put multiple forward and reverse primers to different targets together. The key to multiplex PCR is that all targets must amplify efficiently. But how do you know which target is amplified? So the way to do this, one way to do this is to take the nucleic acid and put it on a gel, wherein once you put it in this agarose gel and you apply electric current on opposite sides, you can see that since DNA is negatively charged, it will move from negative to the positive side. In this manner. So you can see that when you apply the current here, you can see that the DNA will move in this fashion. And if you then design your multiplex PCR to having different sizes, you will get amplicons of different sizes. So you can say, and based on that, you can identify what it is. But for our particular example here, CSF, this kind of assay is not ideal because so many different PCRs will still be required, and it won't be very sensitive enough. So the way to overcome this is by using a fluorescent probe. Now fluorescent probes which are there, each typically have sit between the forward and reverse primer and have a reporter dye and a quencher dye. The reporter dye, when it's hit by laser, gives off fluorescence, which is immediately caught by the quencher. So in its normal unbound state, you can't get any fluorescence out of it. But when PCR takes place, or when this probe is specifically bound, the Taq polymerase comes and breaks this reporter and quencher and separates it from one from the other. And what you then get is, when this is hit by laser, you get fluorescence. So you apply probes which sit between these primers of different colors. So for example, SARS-CoV-2 probe labeled with FAM and influenza probe labeled with X, so by which you can get different colors. So in this COVID testing, what you commonly see is a SARS-CoV-2 which is detected with one color and a control gene which is detected with another color. So this is one example of mutation testing or the use of PCR. The second type is detection of one specific allele. So when you want to say specific allele, you want to know whether one particular mutation is present or not. So these are known as allele specific PCRs. So when should you do allele specific PCR? When the mutation in question defines the disease, for example, JAK2 V617F or BRAF V600E, or sickle cell testing using E6V real-time PCR. So the caveat here is that the mutation must be very commonly present in the context of disease. Panel of allele-specific assays can then be used to screen for mutations, and if they are negative, then you proceed with sequencing. So commonly this approach is used for screening for beta thalassemia mutations. So when should you not use allele-specific PCR? When the mutation in question is highly variable for that gene. For example, FLT3 internal tandem duplication, where mutations across all the gene, where the mutation is highly variable, and sometimes when mutations across all the gene are known to occur for all over the gene. For example, in large genes such as TP53 and so on. So sometimes you can see that these mutations are commonly present at one particular hotspot. So for example, 50% of DNMT3 mutations are R82 mutations. So these particular mutations can be targeted by allele-specific PCR, but the rest of the mutations are scattered all over the gene, and you really cannot target them using this, and you have to use sequencing of that gene. So when you say allele-specific PCRs, they typically have multiple primers. So let's look at DRAF V600E. Here at this particular 1799 position, the G changes to an A. The T is the wild type allele, and the A is the mutant allele. So you use multiple primers here. One is a wild type specific allele primer, and you have a mutant specific primer, and you also have outer forward and reverse primers. So you can see that you have multiple amplicons. One is a PCR that takes place between outer forward and reverse mutant, which results in a mutant-specific amplicon, a forward wild type and a reverse outer wild type, which presents in a more smaller amplicon like this, and then a forward outer and a reverse outer, which actually shows that amplicon. So in a mutant case, you get 2 amplicons, and in a normal person, you get in the mutant case, you get 3 amplicons, and in a normal person, you just get these 2 amplicons. So this is an example of allele-specific PCR. This is how it's done in our lab, and this is how we look at V600E if it's present or absent. So when a mutation is particularly variable, for example, the FLT3 internal tandem duplication. Now it is really important to know for this mutation, because if you look at acute myeloid leukemia, presence of FLT3 internal tandem duplication predicts for an inferior outcome. All right, and these mutations that are there are highly variable, and they are present in this juxtamembrane domain and transmembrane domain kind of mutations that are there. And these juxtamembrane domain mutations are the ones that cause these internal tandem duplications, and you cannot really detect them with allele-specific PCR because the indel varies from case to case. So how do you detect indels like this? So what we do when you have highly polymorphic regions where indels predominate is that you use something known as fluorescently labeled primers and capillary electrophoresis, or also called as fragment length analysis. So in a normal person, let us say for this particular gene, you get a 169 base pair amplicon. If there is an insertion that takes place, for example, a 4 base pair insertion inside of this gene, and you do a PCR, you will get 169 plus 4 bases, that is 173 base pair amplicon. So this capillary electrophoresis allows us to detect small mutations even up to 1 base pair. A classical example of this is the detection of FLT3 and NPM1 mutations. So you can see that this is done on an instrument called as this Sanger sequencer, which allows for fragment length analysis. What you have is you have this reservoir wherein you have a glass capillary here. So you put the sample here and you apply a charge across the capillary, and this sample moves from here all over to this point. At one particular spot, it gets interrogated by laser, and if the dye is present, it gives off fluorescence, which is captured by this detector and then goes electronic processing to form these multiple graphs that you can see here on your computer. So shorter DNA fragments move fast and longer DNA fragments move slowly. And since the time that is required from starting point from here to this point is noted, and if you use a ladder that is basically known standard of different DNA sizes, you can then estimate what is the size of the DNA that has moved across based on the time it took to move from point A to point B. So based on this, you can detect different FLT3 internal tandem duplications. So in a normal person, there is no FLT3 ITD, you have a wild type allele here. And this is an example where you can have a wild type allele with a 360 base pair, that means a 30 base pair insertion, or a 60 base pair insertion in a different case of FLT3 mutated AML. And also another application of this is short tandem repeat polymorphism. So short tandem repeats are actually microsatellites or microsatellite polymorphism, wherein you have small segments of the DNA which multiply. So these typically occur in highly polymorphic loci, and these are inherited from parents. So in microsatellites, you have repeats of 2 to 7 base pairs, which are repeated at a locus. Each of these is known as short tandem repeats, and they are shared between 5 to 20% of individuals. So you can see here that there is a stretch of CAA, which has 4 repeats in one person, 6 repeats in another, and 5 repeats in another person. And if you analyze different STR loci, we can identify the person which is there. So here you can see that you have this ATGC repeat in one person, which is 4 repeats, and in another person it's 2 repeats. So in this person where there are 4 repeats, you get a larger amplicon, and in the smaller amplicon you get 2 repeats. So in person 1, you will have a 4 repeat or a bigger amplicon here. In person 2, you have a smaller amplicon here. And if these were a male and a female and they were to marry, you can see that you will get a mix of these 2 amplicons with 2 different intensities, with different intensities and different lengths of amplicon in this manner. So the applications of the short tandem repeat technology are for paternity testing, for forensic criminology, for post-transplant chimerism testing, or assessing maternal blood contamination in CVS samples, for prenatal testing, where you want to see if maternal blood has contaminated in the chorionic villus, or establishing identity in natural disasters such as earthquake, as we have seen in Turkey now, or burn or a large fire that took place or a crash site where you can't identify the victims. So in such cases, you use these short tandem repeats. So another application of PCR testing is also multiplex ligation dependent probe amplification. So let us look what happens here. Here you have these probes of different lengths which hybridize to target gene, and each of these probes has a stuffer sequence that is present here of different lengths, and every probe has then common forward primer and reverse primer binding site. So if you do PCR, you will get if you do PCR across this, you will get multiple amplicons of different sizes because the length of the stuffer sequence has changed here. And if you use a fluorescently bound primer, all of these will be fluorescently bound. So when you do fragment length analysis here, you will get that different types of size amplicons are here. So this is exactly the principle of multiplex ligation probe amplification, or MLPA technique, wherein you compare the length as compared to normal. So if there is deletion of one particular locus, you will see that the sizes of those amplicons will be less, and you can then predict that copy number alteration is present or absent. So there is BCR ABL like ALL in which these copy number alterations predominate, and you can use MLPA to detect these type of abnormalities. So I think Aniket will speak upon this, but You also do RNA-based testing to look for fusions. Chimeric gene fusions are typically occur inside of introns at a genomic level. But if you were to do a DNA-based PCR to look for BCR ABL, which is taking place between DNA, it would be a very challenging PCR on a routine basis because it would be a very high or a large length of PCR. But if you work on RNA, this would be an easier easier PCR to work with, because as we know, between DNA and RNA, this intervening sequence or intronic sequence is spliced out. So you have RNA which is converted to a complementary strand of DNA as RNA single stranded, and this is done by an enzyme called reverse transcriptase. So when you say RT-PCR, it can be confusing. The real terminology for RT-PCR is just correct terminology for real-time PCR is qPCR. So as I mentioned before, in real-time PCR, how it differs from standard PCR is the use of a fluorescent probe. So what is different between real-time PCR and standard PCR is the cycle at which the fluorescence appears is directly proportional to the quantity of template that is there. All right. So you have different strategies for real-time PCR which are absolute and relative quantification. I won't be speaking of this because I think Divyansh, Aniket will be speaking on these things. All right. So let us move across over to another application of PCR. So which is which is Sanger sequencing. So in a standard PCR reaction, you use these deoxynucleotide triphosphates. When you say deoxynucleotide, you can see that there should be two OHs here. One OH is knocked out, and this OH then removes the H or contributes the H and links to this O to form this new chain, and that is the very basis of PCR, which is facilitated by Taq polymerase. So you can see here that this H acts as a donor, and this O then connects to this O and to form this new DNA chain. But when you use a mixture of DDNTPs, which is the base in which both these OHs are knocked out, and you just have this pentose sugar structure here, and you use a DNTP, if you use a mixture of these, you will get PCR fragments of different sizes. And if they are, if we know which base is added at one particular time point, we can then separate these on an capillary electrophoresis to know the sequence. And this is actually was the principle of Sanger sequencing, when you use a mixture of DDNTPs and DNTPs and separated these at high resolution on capillary electrophoresis. So the disadvantage of Sanger sequencing was that it was very expensive, labor intensive, and if you wanted to do multiplexing, it was very difficult, and low resolution mutations also were not possible to detect. So I'm just going to talk about briefly about next generation sequencing, because this will be talked about by Dr. Srinidhi. So there is a technique that is used in next gen sequencing which is known as library preparation, by which you prepare nucleic acid. I'm not going to talk on this because this is again going to be spoken about by Srinidhi. What is important at the end of NGS is this pipeline, which is essentially use of bioinformatics algorithms to analyze NGS data. And pipelines are very important for NGS or data analysis. Basically, what we do is in a single step, you deliver your FASTQ files, and you get an Excel sheet of analyzable data to work with. So with that, I'd like to say thank you. This is just a brief overview. And yeah, are we going to ask questions now or at the end, Dr. Shweta?
We'll ask later, sir.
All right. So we can, with that, go ahead and with the next talk.
So I request Divyanshu to start sharing his screen and go ahead with this presentation now. Divyanshu, you have to unmute yourself, okay?
Yes, ma'am. Is my screen visible?
Yes.
Okay. Okay, I'll be talking about the topic analytes in molecular pathology and pre-analytical issues. Let's start. So as sir has already told that it's a technique where we detect the DNA and RNA and abnormalities associated with it, and it can be used for diagnosis, prognosis, as well as treatment purposes. And apart from cancer, we can also detect pathogens like virus, parasites, and bacteria and all. It has the technique. It is divided into three phases. It's a pre-analytical phase where the sample selection, collection, and along with everything, the labeling, transport, and till the processing. It is one phase. Next is the analytical phase where we analyze the reports, and then is the post-analytical phase where the clinicians they go forward with our reports. And these are I am showing this as a cycle way, where you can see the first test is issued, then the sample drawing, transportation, and then processing. This is the phase, and it has been seen that 70% of the errors in the report or in the analysis is due to errors in the pre-analytical phase. So if we correct this, then we are going to get 70% of our problems solved regarding the report preparation. So talking about the first thing, the test ordering, means asking for a test. What type of test do we need? First, we need a proper format for the pathologist to know that what he's going to do. So may I ask anyone, any student, anyone who can unmute and tell me, is this actually needed, and what is the real importance of this thing? Can anyone tell me? Or shall I call?
For the patient identity, so this request form should be properly filled because for the patient identifiers, and what type of material was sent, was it a fixed material or a non-fixed material, because that also impacts the process, and the clinical details, whether it is relevant, and all these information should be given. So it is important to fill this requisition.
Yes. Thank you so much. You see, everything starting from the name and address and to the type of sample sent and the clinical details and the clinical history, everything helps in finalizing a diagnosis. So we must have this properly filled, and we should have different variables in it. Like we have to confirm that the sample is coming, then the sample belongs to this patient. So he'll be having the name, will match the case number on that sample with this forms, and we'll also know that what are the tests that have been they want to ask for it, and whatever they are leaving anything, then we can suggest no, this test we can also ask for this test, or if this test is unnecessary, and we can proceed with this test. So we can give our suggestion if we have the clinical history or any kind of data. Okay, next we'll go and talk about the analytes in molecular testing. That is the sample drawing. So now can anyone tell me that what do we mean by analytes? Has anyone any idea? Anyone?
It is a substance or chemical constituent of interest, we can say, like
DNA, RNA, if you have to test for molecular genetics.
Okay, partially correct, but it actually refers to the sample, the type of sample that we can analyze in a molecular testing, because we need samples to find out the DNA and RNA. Then we will go about and ask, and we'll find out if there is a mutation or not, and what are the types of sample that we can process for molecular testing. The sample that contains the DNA and RNA in its natural way, those are known as the analytes. Okay, can anyone unmute and tell me what are these samples? Anyone?
The first one is EDTA blood.
Yes.
Whole blood in EDTA.
Yes.
The second one is Buffy code of testing.
QBC.
And
and
the third one is serum.
yes, the third one is the clotted one. As you told, right, the first one is whole blood. Then the next one, where the plasma and the buffer layer, they have been separated, and the third one is blood serum. Okay, now I'd like to ask you that we are working for DNA and RNA. Okay, so it will be present inside a cell. Can you tell me, is plasma or serum, the fluid part on the top, can they be useful in testing molecular, doing molecular tests? Are they useful? Can you tell me?
The cell-free DNA.
Yes, exactly. Thank you so much. We can obtain cell-free DNA from these samples, and they will help us further in diagnosing cases. And these are a few other samples where we can also do diagnostic testing like swabs, urine test, or bronchoalveolar lavage fluid, and CSF. So we can also perform molecular testing on this, and we can give a diagnosis. And for prenatal molecular screening, we use two types of samples: amniocentesis and chorionic villi sampling. And can anyone tell me, is there a third sample that we can use for this testing?
Abortus.
Quadruplet.
Sorry?
Abortus.
About us, see if we do any test on that, already it's aborted. So we might have any, but there is a simple answer. We can think about it. Simple answer. Where can we find the DNA of the baby? Just before before one slide only you answered that. Okay, no problem.
Quadruplet.
Yes.
Screen नहीं दिखा है.
Yeah, just now it got unshared.
Okay.
Cheek swab can be.
Quadruplet.
Dr. Divanshu, please share your screen.
Is it visible?
Yes, just make it full screen.
Okay, that's okay.
Yes.
So someone said cord blood, that is the right answer, and we can also take mother's blood sample where we can find cell-free DNA of the fetus. We can also use them for diagnosis. And coming to fixed and unfixed tissue specimens, we can see here two diagrams where first one is showing fixed formalin fixed test specimen, and another one is fresh tissue. This can also be used for extracting DNA or RNA. Okay, now we talk about sample collection. And can anyone unmute and tell me what are the problems if we get sample like in these four pictures? What will be the problem? Just tell me what is the problem in the sample? Can anyone tell me?
Diluted. It's diluted.
Which one? The going clockwise. The first, second, third, fourth.
Third. If we go clockwise, the one with the red cap.
Okay. Or anyone else?
First one is in
Is it the
glass tubes,
clotted?
it is clot sample.
Yes. Second one. Okay. Third one. Anyone?
Appendix sample should not be used. It
Yes.
is not
This is
good for
correct.
analysis.
And this one, doesn't it seem inadequate for testing? Because we need to have adequate amount of sample also. So we need adequate sample, unclotted, and there should not be any kind of hemolysis because the hemoglobin that's released, it affects the PCR. And finally, the heparinized sample are also not acceptable. So sample collection, that should be by collecting the problems that can arise. It can be solved if we have the patient and hospital details. The date and time of collection is also crucial, as well as the temperature at which you receive the sample. You must always make a record of that thing. Okay, now talking about hemolysis, there are multiple factors in hemolysis. First, it is the bore of the needle. If you use very thin bore needles, then the RBCs are forced through it and they tend to break. And if you put a tourniquet for a long time, or you extract the blood with too much of pressure, in those cases there can be hemolysis. Or if the sample is transported with under temperature that's not appropriate for the sample, as well as the transport mode. And also after drawing the blood, we should not mix it like that. It's a cocktail. We should always be gentle with the sample. We should mix it slowly and just invert it up and down 3-4 times, then it's done. And even if you get everything correct and you receive the sample in good condition and you still just keep it and you process it in a late stage, in that case also you can have hemolyzed sample. And for avoiding clotted samples, you can have if you have improper mixing, you just pour the blood and just keep it aside. You're not mixing it, or the sample is more than the anticoagulant can handle. In that case, also you can get clotted samples. And inadequate samples in difficult aspirate or dry tap cases, or if a phlebotomist is inexperienced and unable to access veins in a obese person. And as I told, EDT tubes, they are the proper collection tubes. So all these things have to be kept in mind in the sample collection time. For other fluid samples, like saliva for congenital CMV, you must take care that the sample is collected 1 hour after the baby has breastfed, and there should also be some sample from the mother so that we can confirm it that the mother is positive and the baby is negative. And buccal samples you also have to collect by taking attention that the baby has not been fed just now. And in case of swabs, the nylon swabs, they're preferred over the synthetic ones. And then prenatal test, you must have the data about mother's full name, the type of specimen that they're giving, the gamete donation, or any surrogate pregnancy history is there. And
Just one doubt, sir.
yes,
So why should the sample be taken 1 hour after the baby's breastfed?
because you don't want any contaminants with it. The food particles might be there. They will dilute your DNA concentration, and you don't want affecting those things your test, because molecular tests, they're pretty costly, and the things that they're working in micrograms, microliters. So any small amount of foreign particle, it can just ruin the entire test or sample.
Basically, you're taking saliva sample for congenital CMV, so saliva sample from the baby. So breastfeeding will contain maternal cells also, and the
So
maternal
1
cells.
hour enough for preventing contamination.
Okay, shall I continue?
Yes.
Okay. As I mentioned, the type of samples are what container should be taken and what should be the additive in it. And coming to fixed samples, fixatives generally they're used to prevent the autolysis and molecular component decay, but they have also drawbacks. They can cause DNA and RNA cross-linking, which will damage it, and the RNA they will become reverse transcription. They will show reverse transcription inhibition, and whenever the samples they're fixed in Boeing's P5 and Zenker solution, they lead to DNA degradation, and the paraffin that is used, it can cause PCR inhibition. So we must keep in mind all these things before we receive a sample from some center or for any purpose. And then talking about the fixed or unfixed tissue quantity and quality, first of all, we are obtaining DNA from a fixed tissue, then we are taking sections. So we must be aware of what amount of sections should we take so that we can get an appropriate amount of DNA for our test. Like if the tissue is large and covering the block, then we can like 30-40 micrometer section is fine. But if it's small, so we must aim for like 40-80 micron thin slices. And for fresh tissue, 1 to 2 gram fresh tissue is required. And talking about the quality, in formalin fixed tissues, when we are taking sections, they can have contamination. That's the carryover contamination from the blade. If you use one blade for different samples, then they can have transfer of DNA. And can anyone tell me how can we avoid this thing? This carryover contamination from the blade?
If you use a blank block,
Yes, someone, just one person. Someone told the right thing.
so if you use a blank block after cutting
Yes.
each section, so that will take off the fluid.
Yes. Any other idea? Suppose you don't have any blank blocks, then anything you can do about it? We can wash the blade or simply use reusable blades. You can change the blade, right? Okay. Now coming to sample labeling. So it is also a very important part because you can process, you can give results, but unless you don't know for whom is it, you're going to mix up everything. So there can be problems like label failure, where durable labels are not used. You just label it with something undurable, and in the transport only it gets torn out or wasted. So by the time you reach, you have 20 samples, but you have no idea whose sample it is. So everything is wasted. You cannot just process it blindly. And next thing is labeling errors. So you are writing the name of the patient on the tube, suppose, and by mistake due to alcohol swab on your hand, you erase it. And you don't know, and the sample gets mixed up. Again, there is a problem. You cannot just go processing about it. And you should always remember that for labeling, you minimum 2 variables. The more the better. Like name, age, case number. You should have 2 to 3 variables, because if one gets erased out or damaged, then the other 2 can be used to solve the mix-up. Now coming to the transport part, there are several factors you have to keep in mind, because this is very important, because it's not always possible that your lab will be nearby the area where the samples are being collected. Sometimes you have a great lab and you're receiving samples from all over the place. So you must have some guidelines for that. How to transport the samples with the DNA and RNA, they should be in intact condition. First of all, the biosafety and protection, personal protection rules must be followed, and the transport must be rapid, because DNA can survive, but RNA tests they won't survive, because RNA degradation is very fast. And the next thing is there should be sealed transport. They should be packed in such a way that there won't be spillage, and they must be labeled that these are biohazard samples. And as I told, transport must be smooth, so that we can avoid hemolysis. And serum, even if for DNA or RNA doesn't matter, it should be shipped frozen. And plasma and other fluids, they should be preferably shipped at 280 degrees Celsius to maintain the as much amount of nucleic acid possible. And there is another technique that is being used, is dried blood spots. When you dry 50 microliter of blood in a filter paper, and it's specifically good in those areas where you don't have cold chain areas, you can transfer it like that. And stool samples, these are stable for 4 hours at room temperature, but at 4 degrees they can survive 24 to 48 hours. And fresh non-fixed tissues, you should dab them with normal saline soap gauze, so that they don't get dry. And for FFP and slides, you must be cautious that they are not transported without any support. They can break, and the sample can be lost. After the sample receiving, we'll see that how important is the sample storage, because it's not every time possible that you will immediately process it, because there can be shortage of staff, shortage of reagents, so any problem can arise. So you must have some great facility so that you can just store it for few days at least. And it depends on the storage facility, depends on the test required. Suppose you need DNA, then you can store it on 4 to 8 degrees Celsius, but you need the RNA to be intact, you have to have minus 80 degree facility. And you must have always a backup power supply and regular maintenance of your equipment, otherwise you can just lose a lot of samples. And as I told, for DNA, we will be stable at room temperature for 24 hours, and for 72 hours storage, you need 2 to 8 degrees Celsius. And for RNA, you must store them with RNA stabilizing solutions, RNA lighter or RNA inactivators like RNA reagent or DPC, and they must be processed as fast as possible. If not possible, then store them in minus 80 degrees Celsius. The use of glassware should be very cautious. They should be RNA free, and you should avoid multiple freeze thaw. If you are sure about a test, then start it. But you should not be like I'm doing something and then no, you have to not do it, and then you have to again freeze it, again thaw it. That will also reduce the DNA content. And about the fresh non-fixed tissues, they are also with the DNA stable at 2 to 8 degrees Celsius, but you try to store them at minus 20 or minus 70 degrees Celsius for longer duration. And RNA as well, minus 70 degrees Celsius. And talking about the PCR products, once the PCR is done, then suppose in case of like capillary electrophoresis, we first do the PCR and then we dilute it and do the capillary electrophoresis. Those samples, they must be stored a maximum of 72 hours. And if it's fluorescent, then you must store it at minus 20 degrees Celsius, otherwise the fluorescence will be lost. After you have stored it, you have collected it, transported and stored it properly. Now comes the final step about the sample processing. So this is the final step, and if you get it right, then everything is fine then. And the first requirement is that you should have a class 2 biosafety cabinet with you, so that you are because you are dealing with biohazards here. So anyone can unmute and tell me what are these things they are being shown in the diagram, please. Is that regular user using things in the lab? Can anyone tell me?
Micropipette.
Micropipetting.
Yes. Second one.
At the time we can.
Second one. The second diagram. Second photo. Can anyone tell me what it is?
How much quantity you want, you can set that. Like you can adjust that.
No, I'm talking about this one. Can anyone tell me what this is?
Second one is also micropipetting.
No,
This-
this is pipette and this is
Wells.
yes. These are PCR wells and with a sealer on it. They have to seal it so that there is no contamination among these two wells. There should not be any contamination. To prevent that, they use a sealer. And this is a single channel. This is multi-channel micropipettes. So our processing starts with RBC lysate. First, we have to remove the RBC from the WBC. So what we do is use the buffer. The important thing is that we have to prepare it daily. It should be fresh. The pH should be perfect for it to work. Otherwise, the WBC and the genetic material won't be preserved. Next thing is after you lyse it, there will be formation of a WBC pellet. So while supernatant, you must be very much aware that we are not removing the pellet also. Otherwise, all the material is gone and the test is also gone. And the third thing, you must have appropriate amount of blood sample, because if you're not going to process the correct amount of sample, you will not get the correct amount of DNA for the test. And finally, the entire procedure should be done on ice to preserve the genetic material, the RNA mostly. Next, after the pellets are obtained, then we can go for extraction of the DNA and RNA from those. And this should always be done according to the protocol in your lab. It can be manual, it can be automated, but you must follow the protocol and you must not change it unless your baseline protocol is not working out for some reason, or you want to make it cost effective or bring about any changes. You have to follow a new protocol and then only bring it into action. And other problems that arise here are bead contamination. Beads are something which we use. They're paramagnetic beads. Basically, in simple terms, they will go and bind to the DNA, and then the other part, other things will be discarded. Now, this from this DNA, from these beads on which the DNA is attached, we just wash those beads and remove those DNA from them and throw the beads. But while we must be sure that those beads, some few amount of beads, they don't come along with the DNA, because in the further PCR process, they will be also harmful. And also, when we extract a DNA and put it in a DNA container, when we store it at end of use, we must be sure that we are transferring the correct sample, because if we mix up at that time, then our entire result is not going to be matching with anyone. And for extraction in fixed samples, what we need to do is we have to first see that if the sample is entirely from a tumor area, then it's fine. But if it's a big sample and tumor is only on a small area, then we have to first go and see the slide and then find out the area, and from that area only we need to take the sample from the blocks, so that we don't have any kind of dilution, DNA dilution. Next thing is the PCR. After that, we do the PCR. The most important thing for PCR is that you have to have different areas for a stages of PCR, like pre-PCR, PCR, and post-PCR. These areas should be divided in the lab. They should not be doing all the things in one room, because there is high chances of contamination. The first thing, after separation of areas, we need to make sure that our reagents are within the expiry date. All the master mix, the primers, the additives that we use, they're working properly, and the template that we are taking, it should be of right amount and it should be also right quality. So for DNA, like you take a qubit test, and you will tell us that how much amount of DNA is double stranded and that can be sequenced. And for RNA, we do ABL1 copies. This is the gene that continuously expresses itself. So we can do for that all the patients, and you can see that it is actually functional or not. And there can be amplification parameters, like we set in the program in the machine, and there is annealing temperature, there is denaturation temperature, there is the provision of the amplification, all the temperatures they have have to get the perfect time. And finally, the pipetting errors. The pipetting, it should be very skillful. It should not be very vigorous. You are not going to introduce bubbles into it, or you are not going to spill it around. So you have to be very skillful while doing the pipetting. And talking about Sanger sequencing, we will not go in details. Just a few points I will telling that it is of two stages. First we do the sequencing, and then we do the purification. In sequencing, cycle sequencing stage, we have to do it under standardizing conditions, and the pipetting, as I told you previously, because it's basically like a PCR. Only the primers will be different. And after that, the purification is done in here with the help of beads. Now here what happens is the beads, they attach to the other impurities, and they're separated. Like previously I told in extraction.